Hello. We are going to talk about DNA extraction. So, living beings and any other thing are all made up of cells, no matter if you're a flower, a banana, or a human being. Each cell contains DNA inside it, which is a molecule with the necessary information to build an organism. We need DNA to know our inheritance, coding for proteins, and providing instructions for life and its processes. Not only that, it is used as evidence in courts to identify bodies, track down blood relatives, and to look for cures of diseases. This is why we think it is so important to perform DNA extractions, a technique used to isolate DNA in a biological sample. The steps to do so are breaking cells open to release the DNA. The detergent breaks down the lipids in the cell membrane and nuclei. Step 2 is to separate DNA from proteins, and the cellular debris can be removed by filtering the sample. And step 3, precipitating the DNA with an alcohol. DNA is soluble in water but insoluble in the presence of salt and alcohol.
Results in this experiment is in a epithelial test some important compounds that are key elements from the experiment are telos things in our cheek are epithelium cells, the salt and water mixed help consider the fingers we were naturalists the DNA changed and make the molecules less hydrophilic, made it become less soluble in water. DNA is found in everything around us and is shaped like a balloon extracted molecule with a twisted double helix from. And the fruit test measured the fruit expanded the greatest surface area from which to extract the DNA. The liquid salt is added to help break down cells membranes to release the DNA. The filtration step allows the collection of the DNAs and other cells substance. The precipitation step allows the DNA to separate from the cells and substance. The quality of the DNA obtained is small and delicate, but enough of us to provide it.
Summing up everything we said, we learned by doing the experiment that the loose things in our cheeks are epithelial cells. The saliva, salt, and water mixture help us neutralize the DNA charge and make the molecule less soluble in water. The salt also helps us to remove the proteins that are bound to the DNA and to keep the proteins dissolved in the water. The dish soap was meant to break down the lipids and the fats. So by breaking them down, you free up the DNA inside the cells. And finally, when we poured alcohol in the mixture, the DNA became visible since the DNA is soluble in water, but insoluble in the presence of alcohol. Something similar happened with the fruit test. Mashing the fruit exposed a greater surface area from which we extract the DNA. The liquid soap again was added to help break down the cell membranes to release the DNA, and the filtration method allows us to separate the DNA from other cellular substances. And again, finally, we poured cold alcohol to the side of the mixture to allow the DNA to separate from the other cellular substances, because again, DNA is soluble in water but insoluble in the presence of alcohol. It is very important to learn how to extract DNA, since it can help us to further study genetic causes of diseases, for the development of diagnosis and medicine. DNA extraction can also be helpful for detecting bacteria and viruses in the environment. So, yes, DNA extraction is very important to learn.
Well, thank you very much. Bye.